So maybe 2 words about myself. I started out in physics many years ago, did a PhD and initial postdoc in mathematical physics, working on like analysis of fluctuations and Goldstone modes in things like Bose-Einstein condensation and quantum spin systems. But then at some point I felt I wanted to do something a bit more practical. I was hopeless with experiments, so I could never have done the kind of nice things we just saw. And then instead I ended up in bioinformatics and computational biology. But now very recently I've been drifting back a little bit towards physics again. It could be the onset of a midlife crisis, I'm not sure. And basically, so what I'll present today is very preliminary work. It's just sharing some thoughts and ideas of things that I started working on basically 4 months ago when I arrived here for my sabbatical. I have no students yet to thank because I've not dared to engage anyone in things that may or may not lead to something. So it's just some initial thoughts and ideas, and if you have feedback, I would very much appreciate. So just for a little bit of context, we've heard a little bit already about signaling. Once you dive into molecular biology, you very quickly arrive at these kind of things. So gene regulatory networks. These are kind of models or representations of the inner working of a cell. Each of these variables here represents a gene. A gene encodes for a protein, so protein is produced. And then, say if you, for instance, focus on this one, you get a protein, and that protein itself will then affect the rate at which other proteins are produced. And this is how you get this whole cascade of events. They're particularly important in developmental processes, so how you go from a single cell to multicellular organism. But they also play an important role in the kind of signaling that Massimo was talking about. So as a model, they are fantastic. Like you can almost read them the way you would read a computer program. The problem is that this is only one part of one network. And these things take like years and years and years of experimental work just to tease out what are these individual interactions, who is controlling whom, and how does it all work. If it's in development, you can argue we only need one per organism of interest. That might still be doable. But then if you talk about signaling, each of our different types of cells in the body has a different type of signaling. So these networks are all different. And we have about several thousand different cell types just in our own human bodies alone. So then it becomes completely impossible to do this systematically, map these networks experimentally. And so for about 20 years, there's been this idea that maybe can we take a sort of computational shortcut to this problem of mapping these circuits in the cell. And the idea is rather than experimentally manipulating protein by protein, you just observe what the cell is doing under a wide variety of inputs. So it's a bit what in engineering is called reverse engineering. You have a system, you want to understand the inner working of a system, and you just observe how it responds to multiple different types of output, different types of input, and from the output, hopefully, you can reconstruct the actual inner workings. And then you have this kind of various algorithms that try to achieve this result. This is, of course, a typical instance of this kind of shadows on the wall problem, where you observe something which you hope corresponds to reality, but of course you only see the shadows and not necessarily the real mechanism. So I don't think anyone in the field really believes that through this process we would get exactly the accurate maps from the first slide. But at least you would hope that maybe you can narrow down the type of experiments that need to be done, or you can somehow predict how the system might respond to a perturbation, even if you don't know the exact individual details of each and every network interaction. And so like I mentioned, this has been going on for about 20 years. And so we can ask ourselves, now where do we stand after 20 years? One thing that has definitely improved over 20 years is the type and the amount of data that can be generated. So there's been a huge data explosion. But this is just some quotes from two very recent reviews where they had a sort of broader look at this field. And conclusion is basically that this kind of forecasting of what would happen for unseen experiments is not really working very well. So especially here, capture causal relationships from perturbation assays. This is exactly the question you want to answer. So you want to be able to tell an experimenter, if you do this experiment, I expect the cell to respond in this way, even if you don't know the details of the network. But basically, the result is that these methods are almost not able to do this. It's kind of a typical situation of many fields, I would say, right now. There's this famous quote, we are drowning in a sea of data and starving for knowledge. And if you translate that a little bit in what is happening in this field, I believe, is that we are drowning in a sea of methods and really starving for good models. And that's where the physics might hopefully come in a little bit again. So my own thinking in this area has been influenced quite a bit by this paper. Came out a couple of years ago from the group Piani, which some of you might know in the non-equilibrium physics field. It's about understanding causation via correlations and linear response. So to this audience, I don't have to explain what a fluctuation-dissipation relationship is. Basically, it connects non-equilibrium properties, responses to perturbations, to equilibrium properties, so spontaneous fluctuations. Or you can say that in a different way, it tells you what the result of an experiment, a perturbation experiment, will be without having to do the actual experiment. And this is precisely what we are hoping to do in this field of reverse engineering gene networks. So rather than one by one doing the experiment, perturbing your genes, seeing what the effect is, you would hope that you can actually compute the result by just observation, so without having to do the experiment. So how does it work? Many of you will be familiar. So you have a response matrix which tells you what is the average change in, say, a variable, in my case, genes, number k, if you perturb a gene, other variable at time 0. And then the fluctuation dissipation relations, they tell you that you can compute this response matrix exactly from a 2-time correlation function in equilibrium. So you have some variable here measured at time 0, and your response variable measured at some later time. So in theory, if we leave aside the question of whether equilibrium is a good approximation or not for the cells, this is telling us exactly what we need to do our program of reverse engineering. So why after 20 years is it not working? This is something you would, for instance, if you just measure how the cells, you just measure the values, say concentration of proteins in your steady state, and then you just estimate the distribution. So we don't, in a way, we don't know anything. So it's all about estimating from data. So the reason why this has not been applied or not been used in molecular biology is that if you want to do this estimation, you obviously need time series data. And the type of experiments or the type of data you need from your molecular biology, these measurement technologies are destructive. So that means if you want to get the numbers, you have to kill your cells. That's as simple as it is. So no matter what you do, you take the measurement, your sample is lost. And that means that you can basically not follow things over time to get the kind of time series data that you could put into this equation. And so the big question that I try to think about is what can come or what can replace time series data so that we can still try to somehow apply this formalism. And so, this is just an example showing what it's doing. So here is a simple, it's from the same paper, simple simulation where we have 3 variables. So x is controlling y and z. And then there's a back interaction from y to z. Here it shows the correlation between z and y. So the correlation function, you don't see any influence of the epsilon, but then in the response function, which is plotted here, you see that as you increase the value of the epsilon, you get the bigger peak. So it's a difference between correlation and causation from response. But as I was mentioning, so what can replace the time series data? So one line of thinking that I've been considering is that proliferation, so the process where cells divide, that this could potentially replace the kind of time-delayed information that we need in these fluctuation dissipation relations. So as was mentioned earlier, this conference, proliferation is really the sort of a hallmark of what cells do. So every cell wants to divide. Strictly speaking, that's only true for bacterial or cancer cells, but nonetheless, proliferation is really important. And many of you, so most who work in active matter, you might have seen this perspective from about a year ago, which is discussing exactly why proliferation is important and should be studied in active matter. So I think Massimo briefly mentioned we ignore proliferation in the models for now, and Silke also mentioned briefly in passing that we can kind of deal with it. I think what this paper is arguing is proliferation is not something that either you ignore or you do kind of as a side thing, but it's really something that is really of intrinsic interest in active matter research. So I'm not going into any of such complicated business. So what is important for my interest is proliferation basically gives rise to tree structures. And this is something that already was understood by Darwin in, when he first was thinking about his theory of natural selection. So this is a sketch from his notebook in 1837 of sort of thinking about the tree of life. So this is proliferation, not of cells, but in this case of species. But proliferation, this kind of division and evolution, is really intrinsic at essentially all scales of biological dynamics that you might be interested in. So where does the, now the whole idea coming in of why could proliferation tell us something about time? Basically think of a very simple example. So we start with a single cell. It divides into 2 daughter cells, which divide and divide and divide. So you get this kind of proliferation tree. You have a bunch of cells alive at the end. And now imagine if you consider these two, say 2 sister cells. These 2 cells will be quite similar to each other because they have a recently dividing. So for instance, in this case, they share the same mother cell. But if you compare, for instance, this cell to this one, their most recent common ancestor lies much more distant in the past, and therefore these cells will be much more different from one another. It's the same thing as in a family tree where you would be more similar to your brother or sister than you would be to a distant cousin. So it's the same idea here. And so the hypothesis is basically that if 2 cells are close to each other in the tree, their similarity or their correlations would be equivalent or reflect short time delayed correlations. Whereas cells that are kind of distant in the tree, their similarity would reflect long time delayed correlations. So the whole hypothesis is that rather than looking at time series data, so things that evolve in this direction, if we have this kind of proliferation tree, that it's enough to look at one time and just measure the correlations that we see at that one time point, as long as they have the structure of this kind of tree. And so this is the idea that I'm trying to explore, and I'm doing this in the context of data from classical experiments. So It's an experimental design invented in 1940s by Salvador Luria, an Italian, I think he was physicist probably, and Max Delbrück, molecular biologist. So they worked together. And what they were trying to show is that mutations arise in bacteria spontaneously rather than in response to external pressure. And the idea here is basically that sometimes if a bacteria has a spontaneous mutation early in this tree, what is called a jackpot mutation, then sort of all the descendants would inherit that mutation, and such a clone would be potentially highly resistant to then later on challenging by a virus. And basically the two competing theories, whether mutations arise spontaneously without challenge or only after a challenge, would give rise to different statistics, and therefore, if you measure these different individual clones, variation between them, you can learn something about the mechanism that is going on inside. So the data I'm looking is not from that old experiment, but from sort of a modern take on this. In this case, what is done is, so you have multiple clones that are grown from a single cell. So you start with single cells. Each of them is grown to a clone. And then what they observed in this experiment is that some genes, so some variables, show a high degree of variability across these clones, whereas others essentially show unique distribution or same distribution across these different clones. The idea here is basically, if you think of genes switching on or off, if a gene turns on, but it sort of fluctuates very rapidly between on and off, then it switches on maybe early on, but then it switches off again, and there's never any chance to build up a large proportion of on genes. But if the switching between on and off is very slow, then what may happen is a gene switches on early in the tree. Because the switch off takes very long, by the time it's switched off, the whole thing has kind of exponentially proliferated. And so in some clones where the switching on happened early, you would see a very high count of these, or very high value of these genes. In other clones where it didn't happen, you wouldn't see this kind of pattern. This is what the real data looks like. So these are genes, so variables that are what they call non-heritable. So in blue, you see a distribution of like a noise control. So in pink, it's the noise control. In blue, it's just a variation across individual clones. And here you see that some of these genes that are heritable show a very high variance across clones and a very sharp peak in the just the steady state distribution. Whereas here they are actually completely overlapping. So what I want to do is build a very simple model for this kind of experimental setting, and then see if we can use it for the purpose that I started with, this reverse engineering of networks. So what is the model? It's the simplest possible model where we take fixed cell cycles or fixed generation times. So it's like a clock where every unit of time, the cells that are alive at that time, they divide into daughter cells. It's run up to a certain number of cell divisions. And in the end, you have an exponential number of cells alive depending on the number of cell divisions. What matters for us is that if you look at an ancestor that divided at time k, the number of offspring here is again exponential in this time difference. So the longer ago, the more descendants it has. And then the final thing is if you look at any of the given cells that are alive at the end of your experiment.
Sorry. So are you assuming all of your cells survive and that this is neutral mutations, or?
So in in this model, we're not looking at mutations at all. So it's only variation in the activity, so the gene expression of the gene. Yeah, sorry. So,
yes, yeah.
so the original experiment from Lurie and Delbruck was on mutations which may arise and go away again. Here it's fluctuations in the expression that may go on or off. And so what will be important at some later stage is that if you look at any cell that is alive in the end, you ask or you go back to its common ancestor at some earlier time, the number of other cells that share this particular one as the most recent common ancestor is again this exponential factor. So these numbers will come back later on. So the model, I don't want to use this kind of on-off switching model that I talked before. I want to use in a way, a simpler model, a model that I don't have to look at one gene at a time, but can eventually couple all the variables in my system together. So just a very simple Ornstein-Uhlenbeck process. Most of you will know this very well. So you have here a rate at which the particle or the gene is pushed back to its steady state value. The half-life of the fluctuations is log 2 over this parameter. You have an additional variance from random fluctuations, and then the steady state value, which we can set to 0. You will know that the distribution of this random walker, it decays exponentially back to the steady state. So the steady state is where, in this case, we've set the center to 0, and it's normally distributed with some steady state variance. And the steady state variance is just the ratio between the contribution from the random fluctuations and then here this drift parameter. And you also see immediately that it would not be enough to look at steady state distributions because we have, from the steady state distribution, we get one parameter that we can estimate. But to understand the model, we need both parameters. So this is where the basically correlation is not causation comes in. And then the model we use for this kind of proliferation model is a branching process. So the idea is during the lifetime of a cell, it just fluctuates with this Ornstein-Uhlenbeck process. And then when it's time to divide, two new processes are started. Their initial value is the final value of the mother cell, and then they evolve independently. So it's indicated here. So you fluctuate up to a certain time, and then the two daughter cells start with the same value, but afterwards they diverge. And so the correlation here between daughter cells is only due to their shared initial state. So remember, the thing that we want to explore is whether we can say something about time-delayed correlations from this kind of correlations at the end of the process. And in this model, this is completely trivial. So you can compute on the one hand, so suppose you have a single realization of that process, and you check what is the covariance between the initial state and the state at some later time t. And then, of course, it's standard calculation. It gets exponential decrease of the correlation with rate alpha. So over time, the correlations decrease exponentially. Now suppose that you have, instead of one, you have two independent realizations of the process, but with the same initial value. Then we get something that was also used in the talk by Satya a couple of days ago, where these two things are not independent, but they are conditionally independent given a third variable, and that third variable is simply the initial state. So given the initial state, they're independent, you work out, and again, you get the same exponential decrease. But this time, it's not a time-delayed decrease. It's the same time measurement, but of two two random variables that started with the same initial state.
Two different branches.
In two different branches, yeah. And The factor of 2 here is also easy to understand. So if you just follow the process forward, you will say from the black one here, you follow forward 1 time unit for your time-delayed correlation. In this case, if we compare those two, we first have to go back to the ancestor and then forward to the other one. So we double the time. And therefore, we get a factor of 2 here. Now, in the experiment that we have, remember, we don't have access to individual cells at the end of our process. The only thing we can measure is their total fluctuation, so we just measure. So I don't measure individual cell values, I only measure the total value of a gene at the end in a single clone. But then you can work out what this variance is, so the variance of a single clone. And after a bit of work, so you just group together cells that share the same common ancestor with the cell of interest, and the result in the end looks like this. So you can write, or you can compare the variance. So imagine you have, in the experiment, they start with about 50 single cells. Each of these cells is grown to a separate clone, so you have 50 clones. In each of these clones, you measure the total concentration of all the genes. And now you ask, what is the variation in total level across your 50 clones? So you have 50 numbers, and you can simply ask, what is the variance or standard deviation across these 50 clones? There's also a separate measurement where you take data not from clones that are derived from a single cell, but just a random mesh. In the random mesh, variance is simply the steady state variance, or you have the clonal variance, and the ratio of those two is a simple function of this rate parameter. And this is really what I call this fluctuation dissipation relation. So it's again the same story as the classical one. So you have an expression for a non-equilibrium parameter, which is the rate at which fluctuations decay back to equilibrium. And you can express it completely in terms of equilibrium fluctuations, in this case, fluctuations that are measured at one single time point. And then there's this interesting phenomenon where you see correlations, they decay over time. In this case, they decay with respect to the age of the common ancestor. But at the same time, there's the exponential explosion due to proliferation, where if your common ancestor is older, you kind of share more cells that had that common ancestor. And so then there's a balance between this exponential increase from proliferation and the exponential decrease from the decay of correlations. And then depending on the value of this parameter, or depending on its half-life, you see that either one of those two terms is going to win. If the half-life is bigger than, in this case, 2 cell cycles or 2 generation times, then you see this exponential increase in this ratio. So you see that the variance that you compute across clones from single cell is exponentially greater than what you would get as a variance in the steady state. And this is this phenomenon which in this biological paper they called memory or transcriptional memory. So the memory refers here to the fact that the system remembers an early fluctuation away from equilibrium, because you fluctuate away from the steady state, and then there's this explosion due to proliferation. And if the fluctuation goes back to the steady state slow enough, then it kind of the whole clone remembers this initial fluctuation. But for our purposes, it basically means if you can estimate those two numbers, you can estimate this parameter, and you don't need any time series data for it. So this is where having this phylogenetic tree information becomes equivalent to having time series data. I won't go through all the details, but you can essentially do the same thing with, with a multivariable process, where now we have a matrix that connects the relationships between our variables in the system. And what you find is that if the underlying process is reversible, that's something we can discuss later, essentially you get the same information, but now it's a matrix equation where we have the matrix of coefficients in which we're interested, and we can express it completely in terms of two covariance matrices, which we can compute directly from our data. And so again, we observe this memory if one of the eigenvalues is greater than this two-cell cycle. So I did some very simple simulations, so just a system where things go back in one circle in both directions, just to check whether this thing actually works. And the sort of, in the end, not very surprising, but initially at least a little bit disappointing result. What I'm showing here is the different normal modes or eigenvalues of the system. Here you see the length, the duration of the experiment. So they're sorted from smaller eigenvalues to longer one, or from larger half-life to smaller ones. And this is the overlap between the true eigenvector and the estimated one from a finite number of samples. So if you use estimated ones, there's some stochastic error introduced. And this actually means that you can only estimate the kind of slow modes accurately from the data. The ones that are sort of faster fluctuating, you don't get them exactly. So the equation mathematically is exactly invertible. If you put in the true covariance matrices, it gives you the true A matrix. But once you put in finite sample errors, then only the slow modes can be recovered. But just very briefly, I think actually the slow modes is all I need, because what we're interested in is this, not this exact function, but rather the integral of the function, which can be expressed as the inverse, which gives you the, which is dominated by the slow modes. And just to conclude, I'll skip the, I'll just say that I've started trying this on the real data. Some of the results that come out match what is known about biology. You also get nice sparse networks. So it seems that with this approach, at least something interesting at the beginning of a nice model might be coming out. So just to summarize, I've shown you how, if you use these branching processes, you can derive fluctuation dissipation relations that don't involve any time delay, but just same time measurements in the kind of, at the tips of a tree. It's possible to reconstruct the causal links, at least for the moment, if the dynamics is reversible. And what is needed now is just a lot of extensions of the model. For instance, the model where you assume that all cells divide exactly at the same time is completely not realistic. You want the cell division time to be, again, some stochastic variable, maybe with some exponential rate. So that's something to work on. And then what I didn't discuss at all, but what I'm really interested in is the question of spatial correlations. So there's an interesting phenomenon that when cells divide, their daughter cells actually stay in close proximity. And that means the trees that I discussed, I just presented them as kind of time trees, but they're actually, in the real system, space-time trees. And so it would be interesting, and this is a figure to conclude. So what I'm hoping to achieve is, this is what you do in physics. You generate time series data, have your fluctuation dissipation relations. This we cannot do in biology. Instead, we can get some of this information about proliferation, where it is possible to do experiments. But if we can now extend it to spatial correlations, based on this fact that when cells divide, they kind of stay close to each other, that would be enormously helpful, because as was mentioned already, these days measuring spatial data has become very easy. So that's kind of where I'm hoping to go next. Sorry if I went a bit over time.
Okay, thank you. Questions?
I'm just a little bit confused because I was expecting, I'm not an expert in the field, but the gene network to be highly nonlinear. So the fact that then you're trying to do with the linear approach, so do you really able to capture something, or there is, right?
So, I mean, linearity is obviously approximation. I mean, even just that we're assuming that the states are normally distributed. That's also a big approximation. We know it needs to be more kind of Poisson distributed. But it seems, at least when you apply it to the real data, you do recover some things that are known. Of course, if you compare it with like a real perturbation experiment, and say the real experiment predicts there are 40 targets, maybe you predict the strongest 5 to 6 maybe correctly. So it's obviously a limitation. Yeah.
Tom, I had a clarification. So you said that the correlations, right? They come from two different branches, right? But then I think you explained it where both of them from the same branch.
So it comes. So if you take any two, say, endpoints, the correlations are due to the time when they diverge from each other.